Hello, my name is Jeff Saucerman, and in this video, I'll be introducing you to a software package that we've developed called Netflux. And so Netflux can be really useful for developing computational models of biological networks, even when you really don't know any programming and are really like a biologist or a student or a physician. And so we find that with this software, people without that computational background can build very productive models and gain a lot from them. So this is a little schematic of our very first computational model using this logic-based approach to developing biological networks. And so in this network right here, you see the beta-adrenergic receptor, one of the classic G-protein coupled receptors, that's binding the hormone norepinephrine, and that stimulates a signaling cascade that propagates through a variety of different pathways to increase the contractility of the heart and its hypertrophy, things like that. And so one of the challenges is in trying to model these sort of networks to gain an understanding of how they work as a system rather than just individual proteins. We need new powerful frameworks to do that integration that does not rely on knowing the rate constants and concentrations of everything in this network, which is just not feasible. And so we developed this logic-based differential equation approach where without knowing all of these parameters, we can make predictions about the dynamics by which a network responds in response to various different experimental stimuli, whether the kinetics right here or concentration responses here, look at what's more sensitive than other things. And one of the key aspects here is we're using what we call default parameters, where we assume the certain relationships between the proteins and then test what that can tell us. So the first time we developed one of these models, we were really curious how it might compare to a fully detailed biochemical model that's based on full biochemistry, not just the reaction mechanisms, but also concentration and rate constants. And so we ran a series of simulations where we knock out each member of the network and see how it affects every other member of the network. And so this gives us a pretty comprehensive view of the functional relationships in a system like this. So this is on the right is the original schematic showing the different parts and interactions that are within that network. And so this relied on an awful lot of going to the library. I spent many weeks in the library trying to pull out different parameters that might have been measured like dozens of years ago. And so in comparison to that approach was extremely labor intensive and could only ever really be done for the most characterized networks like beta-adrenergic signaling. We used this new approach called logic-based modeling. And using this, we found without using any of the parameters from the literature, just knowing the structure of the cartoon I showed on the last slide, we can now predict a lot of the things that are happening in the network that are at least qualitatively consistent with what was happening in the full biochemical model. So give you just like a little glimpse at some of the math underneath the hood. We're just, in this case, looking at how A activates C, and we assume that it does so on a saturating sort of relationship like this, saturating curve. That's very common in biology. We're going to assume that rather than looking in, say, like nanomolar units or something like that, we're just going to assume like a fractional unit that's normalized on a 0 to 1 scale, but it's continuous. And then from there, we can put these sigmoid functions that are called like f-act in this case inside differential equations. And all this means is that this is telling us how does C change when A changes. This is saying that when A increases, C will increase. And then this is saying when left on its own without A, this little term right here is saying that C will decrease, so get degraded, for example. So that's the basic idea. Again, we use these default parameters that are shown down here. When pathways come together, then we use what are called like AND gate logics or gate logics. So AND gate would mean that like both are required, and OR gate would be like either pathway is sufficient to allow activation downstream. So this is now a diagram of the first sort of larger scale network model that was enabled by this approach. And so in this, we're showing, we're going to stimulate with phenylephrine right here. And what it does in the cell is it stimulates a gene regulatory pathway that induces hypertrophy or growth of these cardiac myocytes. So this will show that how it propagates information throughout a cascade. And then we kind of wash out the phenylephrine and then add TNF alpha and see how it affects the network in a different way and in a more inflammatory way. Here is another example. This is now our model of the cardiac fibroblast. And so we're adding TGF, growth factor TGF beta, and you can see how the information kind of propagates down the entire network. A really important point here is that we're running simulations of everything in the network at once. You get a very global view from this. And you can also run simulations of any perturbation that you would want. You could do 2-way, 3-way, 5-way combinations, no problem at all. So now let's kind of turn to how you might be able to start to do things like this. And so this is with our software package called Netflux. It's a user-friendly software for doing this logic-based network modeling. And so rather than having to code it all up, we couldn't have coded up ourselves the sort of models that I just showed you. We use this package where we define the networks in an Excel spreadsheet format. And so taking like this example right here, this is showing that A activates C, activates E, that there's a positive feedback from E to C, and that B to D inhibits activation of E. And so that's shown right here where we got different inputs to A and B. We have A goes to C right here. And we have right here, this is an important one to note that this is saying C and not D. So that's showing that they're coming together in an AND gate logic, where if D is activated, that's sufficient to block the actions of C. So that's a common thing. And then this is just kind of a screenshot from the graphical user interface. As you can see, no coding. Basically right here, you're selecting which species you want to plot in the simulations. There are various different, we're called species parameters. So things like this tau is like how fast, like A in this case, will respond to a stimulus, or Y max is the expression level of that. And so if you want to knock out the protein, you would set this Y max to 1, to 0. Or if you want to overexpress it, you might set it to 10. And then you have reaction parameters. That's, for example, how does A affect C? And so you've got a reaction weight like this, where if that weight is 1, then that means that A can fully activate C. But if it's 0.5, as shown right here, then that means that a full activation of A only half activates C. So that's the basic idea. So a few different features of the software package. We've tested it with a variety of high school students, biologists, and physicians, and that really need no programming experience to use. Importantly, our native format for building these models is Excel. So that allows collaborative editing and allows you to annotate your models with lots of literature so that you can make sure that it's very rigorously performed. We can import a variety of different network formats from other modeling packages. So if you want to test out, if you're actually more specializing in a different, like a Boolean format, you can test your model out in Netflux as well. Or if you're generating simulations or generating models in Netflux, you can export it in these formats right here to network graphics packages such as Cytoscape. If you're just going to only use Netflix right here, you'd want to use the CSV export so that you can export the results of the simulations that you ran in Netflix. Or if you want to really get into MATLAB or Python coding to run more advanced simulations, this allows you to automatically generate all of the differential equations and accessory files so that you can use them on your own. And finally, we provide a variety of tutorials and style files for visualizing your network in Cytoscape, which can be really helpful for seeing how a network is functioning. So this package is available freely on GitHub through Netflux. And so we've got Windows and Mac installers and also MATLAB code. So there are a couple of exercises I encourage you to work through. We're also probably going to be posting some tutorial videos on them, but this is kind of set up so that you can kind of explore on your own and test things out. So in this exercise 1 slide, I'd like you to open up the Netflix documentation and Netflux, open up the model corresponding to the simple little network right here, and run some simulations. First, just simulate adding A and like seeing like, well, what does that do? And importantly, I think you should make a hypothesis of what you think it should happen, and then test that hypothesis by running a simulation. Because really, when there are discrepancies between your hypotheses and the results, that's where a lot of learning happens, that might not happen otherwise. And then once you've done that, run more complicated experiments, such as the one right here. So this is showing that we're simulating the A comes on, then A comes off, B comes on, B comes off, and then A and B come on. So once you get through that, there are a few additional exercises you might want to try. One is kind of in more free form to test how changing various different parameters like expression levels or time constants affects the result. You can also try experimental designing in this framework. So you can design an experiment to block activation of E and see if the network responds the way that you predicted. A next step that's important is to try to modify the network by adding new reactions, saving the changes, and then reloading the model. And so you can see whether you can extend the model in that way. Finally, once you've been able to run those sort of simulations, I encourage you to look at the export features, such as exporting the data of the different simulations, exporting the MATLAB files. Even if you don't do MATLAB coding, take a look at those files because it generates all the math behind the scenes, and so that code can be revealing. And then also, it's really fun to do network visualizations in Cytoscape using the XGMML export. And so once you've done those sort of things with this simple example network, then I encourage you to make your own model, just to make something really simple, like 3 to 5 nodes of a biological pathway of your choice. And from a lot of experience, we've seen that students and biologists can get something together in just a couple of hours that is enough to learn even more about the pathway that they may be exporting. So finally, in terms of installing Netflux, so as I mentioned, Netflux is available on GitHub. Please look carefully at the Netflux documentation, and we have installers for Windows and Mac. And then the MATLAB code version of this is also available through the GitHub site. And if you are more proficient in MATLAB, kind of looking at the code of how Netflux works can be further revealing. So thanks for tuning in, and we hope you got you enticed to try out Netflux, and please let me know what you are able to do with it, and keep in touch. Thank you.